So today, our session is titled Every Mom, Extracellular Vesicles as Biomarkers of PTSD in Pregnancy, and our presenter today is Ashley Russell. Dr. Russell is an assistant professor of biochemistry and molecular biology in the School of Science, Department of Biology at Erie, the Behrend College. Dr. Russell is well connected to the CTSI. She's a past CERC fellow and also part of our affiliates program. The affiliates program seeks to officially recognize and formalize the efforts of faculty, scholars, and research staff contributions to the CTSI. The program supports clinical and translational researchers at all of our locations and fosters networking to conduct high quality collaborative research. I do have a short YouTube video to share about the program before Dr. Russell takes the stage. Our CTSI YouTube is full of informational videos of past TSS seminars and many of the other offerings in the CTSI. I'm going to put the link to our YouTube in the chat. Okay, so now I'm going to share this video, and I did practice, so it should work. Okay, let's see if somebody could.
A critical part of Penn State's CTSI mission is the engagement of our communities. The affiliates program is the living embodiment of community engagement.
So the benefits from a staff perspective are really the networking opportunities that come with becoming an affiliate. So connecting with other research staff at Penn State campuses and departments outside of where that staff member currently works, and also connecting with research faculty who may work in areas that align with the interests of the staff member. So really opening up networking opportunities for our research staff.
Our members will be able to apply for CTSI pilot funding mechanisms. They will have networking opportunities to connect with other faculty members to apply for interdisciplinary research grants through Penn State as a whole. We look to create that community of researchers so we can have in-person interactions and connections that are often missing in clinical and translational science, particularly when research becomes siloed.
The Penn State CTSI Affiliates Program welcomes applications from faculty, research staff, and postdoctoral scholars from all Penn State campuses who are conducting clinical and translational research. Affiliates can stay in the know through the SharePoint page that we have dedicated for the Affiliates Program, our Teams channel, as well as signing up for the CTSI newsletter.
So that was a little bit of information about the CTSI affiliates program. I will put the information where to find more information about the program into our chat. And with that, I'll turn it over to Dr. Russell.
Thank you. Hello, everyone. Like Jessica, I also practiced with screen sharing beforehand, so we'll see if it works. Okay, looks good? Awesome. All right. So, hello. The title of my talk is Every Mom, Extracellular Vesicles is Biomarkers of PTSD in Pregnancy. So one of the things Jess and I were talking about before we had everybody join was the way that things were marketed. So the title EV should have been capitalized and it wasn't, but you'll see why as we start to kind of move through the talk, why the EV is such an important piece of the title. So an outline of what I'm going to be talking with you guys about today is first, I'm going to give you some background. I'm going to be talking about trauma and post-traumatic stress disorder or PTSD. I'll talk about pregnancy and then extracellular vesicles or EVs. And then we'll go into the study design and methods. And then we'll talk about some of the preliminary progress that we have made with this project so far. And then our short and long-term project goals, and then future directions for this project as a whole. So when we're talking about trauma and post-traumatic stress disorder, it's important to note that these are two kind of separate but interrelated things. So when you're talking about trauma, you're specifically talking about the exposure to an actual or threatened event, whether that be something like a death or injury or violence, and that violence can be physical or sexual in nature. When you're talking about post-traumatic stress disorder, however, this is where you develop a chronic, debilitating psychological disorder that can occur following the exposure to a traumatic event. So importantly, not all trauma exposures will lead to the development of post-traumatic stress disorder. There's a number of factors that can impact the risk of going on to develop PTSD. That includes biological and psychological vulnerability. So there can be genetic differences that may predispose someone to be a little bit more susceptible to developing full-fledged PTSD. There can also be the confounding variable of having a pre-existing mental health condition, so something like anxiety or depression prior to or coinciding with that traumatic exposure can increase someone's likelihood of developing PTSD. The trauma itself can also impact whether or not someone develops PTSD. So the type of trauma, the severity of it, the frequency of it, the timing in the person's life when it happened. Was this childhood trauma or was this something that happened last Saturday, right? So there's a number of factors that can influence this. And then whether or not the person who experienced the traumatic event has strong protective factors. And when we talk about protective factors, we're typically referring to things like coping mechanisms and social support. Does this person have the necessary skills and support in place to help them navigate the exposure to this traumatic event to hopefully prevent them from developing PTSD? So pregnancy is a critical window for maternal and infant health. Fetal development is highly sensitive to maternal physiology, and adverse maternal experiences can impact both the mother and the child. So things like stress, trauma, psychiatric disorders can affect, again, both maternal and fetal health. And the impact on fetal growth and development can be pretty substantial, and there can be long-lasting effects that go all the way into childhood, adolescence, and adulthood. So understanding how these psychological disorders and impacts on fetal health kind of all work together are really important because they have long-lasting intergenerational effects. So stress biology can play a major role in pregnancy outcomes. So when you are stressed, as you would be if you had post-traumatic stress disorder, you have activation of the sympathetic nervous system and the HPA axis. So your body is constantly going into fight or flight mode, which is not great. And then that can lead to potential hormonal imbalances, immune dysregulation, and vascular changes, all of which can negatively impact pregnancy. So women in general are disproportionately affected by affective disorders like depression and anxiety and post-traumatic stress disorder. The lifetime prevalence of developing one of these psychiatric conditions is double in women compared to men, so about 13% in women and just around 6% in men. And when we look specifically at expectant mothers, we see that 60% of pregnant women experience at least one traumatic event within their lifetime. And of that 60%, 2/3 of them are reporting multiple traumatic experiences. So it's not easy to be a woman, right? And then when we're talking about PTSD rates specifically in pregnancy, we see about 21% in community-related populations, so just the general public. And then when we're looking more specifically at high-risk populations, that number jumps up to 43%. So a large proportion of women can go on to develop PTSD. So PTSD and pregnancy are not a good combination. There are biological mechanisms at play that can lead to adverse pregnancy outcomes. So when you are experiencing PTSD, you're experiencing chronic stress. So like I mentioned earlier, you have HPA axis dysregulation, elevated cortisol levels, you have chronic immune activation and inflammation, which can impair placental function, and changes in the vascular and endocrine environment throughout the body that can also contribute to pregnancy complications. Aside from these just underlying biological changes that can occur during PTSD, it's also typically associated with changes in behavior. So increases in smoking, alcohol, and substance abuse are all pretty common when it comes to PTSD. Poor nutrition, disrupted sleep, and then reduced engagement in prenatal care. All of these things kind of converge into this really not great recipe for negative pregnancy outcomes. So when we're talking about those adverse outcomes, some of the main ones that kind of come into play are preterm delivery and low birth weight. Neither of those are super great. And when we break things down and look a little bit more specifically about the adverse outcomes to the mother versus the children, we see that mothers who are experiencing PTSD during pregnancy have higher rates of going on to develop chronic PTSD and affective disorders later on, like after they have their babies. They also have increased risks of developing cardiovascular and metabolic diseases. And then there are also challenges with these women in bonding with their babies, which can lead to kind of a vicious cycle because it can be pretty traumatic not to be able to properly bond with your baby, right? And so that's going to feed into your PTSD and then make it even harder for you to bond with your baby. So it's a really bad situation. And when we're looking at the babies, we can see neurodevelopmental impairments, or at least higher risk of this. They have higher risk of developing affective and behavioral disorders, metabolic disorders, asthma, immune dysregulation, and these effects can persist into childhood, adolescence, and adulthood. So understanding how to kind of identify these women early on in their pregnancy and find interventions to help regulate their stress levels and cope with whatever their trauma exposure was to treat their PTSD is really important and has long-lasting multi-generational impacts. So extracellular vesicles, or EVs, could potentially serve as biomarkers and potentially mediators of maternal stress. So, like we've just been talking about, PTSD is linked to poor pregnancy outcomes, but we don't understand the underlying mechanisms of what exactly is going on there. And we need accessible mechanistic biomarkers to identify these risks early on. Speaking from experience, when I was pregnant with both of my daughters, when I would go into the clinic in my earlier appointments, I was given like screenings for anxiety and depression. And I remember in my second pregnancy, no one even asked me for my paperwork, and I ended up taking it home with me. So the mechanisms that are in place to identify women who may be struggling with mental health disorders are not super great in general. And I'm talking with friends, this is not a unique experience. So blood draws are things that happen at all of your first trimester appointments, or at least they should. And so within that, there are potential biomarkers that we could pull from so that we don't have to necessarily probe women for self-report information about their mental health status. Kind of the goal here. And that's because these extracellular vesicles that are present in blood can carry molecular signals of stress. So let's take a second to just talk about what extracellular vesicles are. They are the coolest thing I've ever encountered in science, and I love to talk about them. So here we go. They are very small membrane-bound vesicles. They're about 50 to 500 nanometers in diameter, so they're really, really, really tiny. They're released by all cells, and they're present in all biological fluids. I have not come across anyone assaying a biological fluid and saying there are no EVs here. They're in blood, urine, saliva, tears, breast milk. People have found them in the synovial fluid of joints. They are everywhere. And they come through two main biogenesis pathways. So if we take a look at this figure up here, we see a cell This would be a multivesicular body. It was previously an endosome. The luminal membrane of that endosome butted inwards and created these tiny little vesicles inside. So we now refer to this as a multivesicular body. This multivesicular body can now fuse to the plasma membrane and release all of these tiny little packages into the extracellular space as exosomes. Another route of biogenesis is these vesicles can just bleb right off the plasma membrane of these cells, and we refer to those as microvesicles. So there's not a really good way to differentiate between these two populations of exosomes and microvesicles. So as a field, we typically just kind of clump them together and refer to them as extracellular vesicles or EVs. And these EVs contain proteins, nucleic acids, and lipids that are reflective of the cell that released them. And that's really important because they can also mediate cell-to-cell communication. So if one cell releases these extracellular vesicles and the cell is in some sort of inflammatory or pathological state, the molecular signature in the vesicles that are released from that cell will theoretically be different from a normal healthy cell. Another cell can take those vesicles up and the proteins in there can actually function in that recipient cell. The messenger RNAs can be translated into proteins that are then functional. If there are microRNAs, they can repress protein translation in the recipient cell. So there can be really big effects in those recipient cells when they take up these pathological EVs. Another really cool and important aspect of these extracellular vesicles is that they can cross barriers that many things cannot. So they can cross the blood-brain barrier. So if you have any sort of neuroinflammation, things from your brain, EVs that are derived from neurons, could pass the blood-brain barrier and get into your circulation and be taken out in a blood sample. We also see crossing of extracellular vesicles across the placenta. So you could potentially take a blood sample from a pregnant woman, and I don't know if anybody has done this, but theoretically, you could then look at EVs that were specifically made from the developing fetus. So there's a whole bunch of potential with these extracellular vesicles. So because you can just kind of take them from a biofluid and assay them, they have tremendous promise as non-invasive biomarkers for a number of reasons, right? So they're easily accessible. They can be collected via routine blood draws. You can even take a urine sample. Those are very easily obtained in the clinic. They're very stable because they are protected by a lipid bilayer. The cargo inside are not going to be strong targets for enzymatic degradation, so they are more stable and theoretically more reliable than other free circulating RNAs or proteins. They're also quite specific, so the cargo inside of these extracellular vessels can be cell type or tissue enriched. We see a lot of placental-derived extracellular vesicles in maternal circulation that can be separated out from other populations of EVs in the blood. They're also mechanistic because they can actively alter recipient cell function. So looking at what is in these extracellular vesicles can lend insights into what might be happening maybe on a pathological or a physiological level inside of the person who these EVs were derived from. And they're dynamic. So these profiles can change very rapidly in response to biological and environmental stressors. So they can change quite drastically if someone is experiencing some sort of pathological insult. And like I mentioned, they are also barrier crossing. So they can cross the blood-brain barrier and the placental barrier. So we can garner insights into maternal fetal signaling. So as far as I've seen, there's only one PTSD-related extracellular vesicle study, and that was conducted in veterans, so a very different population than pregnant women for the most part. And they did see changes in microRNA signatures in the veterans that were experiencing PTSD versus the ones who were not. So there is a lot of promise here with these small, non-invasive vesicles to be biomarkers. So throughout pregnancy, the placenta releases extracellular vesicles into maternal circulation, and the quantity increases throughout the duration of pregnancy. And these EVs can modulate maternal and fetal physiology. So this can alter immune tolerance to ensure that the mother does not reject the fetus. We can also see changes in vascular tone, metabolic adaptations, neurodevelopment. There's a lot of roles that these EVs have been shown to play. And again, there's that bidirectional communication. So EVs from the mother can cross the placenta and impact the fetus, and fetal-derived EVs can cross the placenta and then be detected theoretically in the maternal circulation. So there's this potential to capture stress-related signals during pregnancy and beyond. There have been recent animal studies showing that in pregnant animals that were experimentally experiencing prenatal stress, there were changes in their EV profiles, and subsequent experiments showed that there were changes in the way the fetuses or cells derived from the fetuses would take up those vesicles. So if it was from vesicles derived from stressed pregnant dams versus non-stressed dams, there were differences in the way the vesicles were taken up. And then there are also a number of groups who are looking at breast milk derived extracellular vesicles. One study in particular showed that there were changes in breast milk derived EVs from stressed mothers versus non-stressed mothers, specifically the microRNA profiles. I also know of some groups that are looking at how breast milk derived EVs are taken up in a fetus. So they will do studies where they label these vesicles, orally give them to a baby mouse, obviously not like a human baby, and then they image them and they see that there's a lot of uptake actually in the brain, which is really interesting to see that milk EVs are impacting the brain of the offspring. So a whole lot of really cool potential there as well. So the hypothesis for the project that I'm working on right now is that PTSD alters EV-associated extracellular RNA, xRNA, profiles in expectant mothers. So for our pilot study, we aim to recruit 20 first-time mothers during their first trimester of pregnancy, hoping to have 10 control and 10 PTSD trauma-exposed participants in the project. We collect blood and separate the blood plasma during the first and third trimester, and we will be doing downstream separation of extracellular vesicles and then subsequent analyses. So we're planning to do EV characterization and small RNA sequencing. And then also at the first and third trimester, we are conducting clinical interviews and a battery of various questionnaires to get a very rich data set about the mental health status of the women participating in this study. So for this project, we are partnering with McGee Women's Research Institute. A few years ago, they set up a campus. Their main one is in Pittsburgh. They set up a campus in Erie, and so we've been able to partner with them for this project. And we consent women who are eligible for this study and determine whether or not they have had prior trauma exposure. And some of our exclusion criteria here are prior pregnancies. So we are just focusing on first-time pregnancies. There is a very different experience for people for their first pregnancy versus their second, right? There's a lot of unknowns that come with being pregnant for the first time. And so that's what we're more focusing on, right? And there's a whole other layer of complexity when you have another child involved that you're taking care of in a second or third or subsequent pregnancy from that. And we also wanted to exclude women who had had any previous miscarriages because they would probably be experiencing much higher rates of stress moving into this now current pregnancy, thinking about their prior pregnancy experiences. So that is one of our main exclusion criteria, is that participants have to be at their very first pregnancy that they are aware of. They also cannot be smokers or substance users. Those things have been shown to modulate EV profiles and would be very confounding for our studies here. And then we also exclude women who have pre-existing metabolic and cardiovascular conditions because those things have also been shown to impact EV profiles. And we want to focus as much as we can on the mental health aspects of things. So then our clinical interviewers are made up of the Behrend Masters in Clinical Psychology graduate students. They've been the ones to do all of the clinical interviews and administer the questionnaires and help with all of the data management for that. So the data that we will get from the participants include our clinical interview data, a battery of self-report measures, and then select data from the electronic medical records. So with the clinical interviews, the graduate students are administering the mini international neuropsychiatric interview to assess lifetime and current psychopathology. They're also administering the clinician-administered PTSD scale and the Hamilton Depression Rating Scale. In addition to the information gathered in those clinical interviews, the participants are also conducting a battery of self-report measures, including basic demographics, medical history that includes both physical and mental health, as well as a number of different measures looking at trauma, PTSD, affective disorders, resilience, cognition, a whole bunch of different things. So we have this really broad repertoire of information about the participant. And then we're also going to be able to pull pregnancy and delivery related health information. So did this woman go on to develop preeclampsia during her pregnancy? Was she delivering at term, or did she have preterm delivery? What was the baby's weight? Were there any complications during delivery? Those kinds of things. So a whole bunch of data that we'll be able to have access to at the conclusion of the study. And then in terms of our biological samples, we are collecting 20 mils of blood at both the first and third trimesters. We separate out the blood plasma and then store that at -80 degrees. And then we will separate the extracellular vesicles via size exclusion chromatography. So if you're not familiar with this technique, you basically have a column that has a bunch of tiny little beads inside, that really, really small. You put your liquid sample on the top of the column and the sample moves through the column. And I always explain this to other people as Plinko from The Price is Right? The best game ever. You put your Plinko chip on the board and it just kind of ping pongs down. And that's what the extracellular vesicles are doing as they move through this column, is they just kind of ping pong back and forth through the column and then they elute out at the bottom. And so the way that this works is the samples will move through the column and the beginning is just void. There's nothing in it. It was just the liquid that was in the column when it was stored in the refrigerator. Our extracellular vesicles are quote unquote big. I mean, they're really small, but they're bigger than the other things that could be in the sample. And they ping pong down pretty quickly. And then they separate out as our extracellular vesicles. Blood plasma has a ton of other stuff in it, like extracellular proteins. These are smaller, right? And so as they move through the column, they get stuck. And then it takes a little bit of time and then they get unstuck and then they move again. And then they get stuck and then they move again and they get stuck. So it takes a lot longer for them to elute out of the column. And so at the end of it, the first 6 fractions of 500 milliliters of liquid are just void. There was nothing in there. The next 4 is where our extracellular vesicle population is mostly concentrated. And then the last 4 is where our extracellular proteins are concentrated. So we can look very specifically at our blood samples or our blood plasma samples and assay, was this something that was in or associated with an extracellular vesicle, or was this just circulating kind of in the maternal circulation? So once we get our extracellular vesicles, we run it through the pretty standard EV characterization protocols. The first is our nanoparticle tracking analysis. So this is just a standard thing that's done in the field to determine the overall size of the particles and how many there are. And then we perform transmission electron microscopy to visualize them. So again, these extracellular vesicles are really, really small. We can't use regular fluorescence microscopy or light microscopy to see them. So we use an electron microscope. The scale bar is set to 100 nanometers, and these would be our extracellular vesicles. So they're really small. And then we do some Western blotting just to confirm that we do indeed have our extracellular vesicles in our sample. And then we're going to perform small RNA sequencing to look at that extracellular RNA. And again, we can do that with our extracellular and protein fractions to be able to compare, is this change in RNA signature something that's associated with a vesicle or maybe a protein complex that's just circulating freely in the maternal blood. And so that can allow us to make those comparisons with EVs versus proteins. We also plan to make comparisons between obviously our control and our trauma-exposed PTSD samples. And then we also want to compare across our first and third trimesters. So one of the important things that we do in those clinical interviews is ask them if there have been any major life events that could contribute to the development of PTSD. So if they were in our control group, went through a traumatic experience during their pregnancy, that's something that we need to know because it could change the basis of where they are within the groupings of this project. So what we have done so far, reminders, we had a goal of 20 participants for this pilot study with 10 control and 10 trauma exposed. As of the 15th, we have 14 participants enrolled, but 4 of them have withdrawn. So officially, we have 9 trauma PTSD exposed participants and then, 1 trauma and 9 control. We had enrolled 2 trauma, 1 of them had withdrawn, and then we did enroll 12 control, but 3 of them withdrew. And some of the reported reasons for withdrawal was that they were changing providers, so they were no longer with McGee. They changed to the other hospital system in the Erie. They moved out of the area. 1 just no longer wanted to participate. And another, which we found maybe could be a red flag, is that their partner wanted them to withdraw from the study. So there's not much that we can do about that, but we are slowly moving forward with recruitment. We have been a little slow with this, and from talking to the clinical staff at McGee, there's a number of issues that are kind of making this a little bit more challenging. And the first is that the overall number of pregnant women presenting is lower. There's less people having babies, maybe something to do with the economy and the cost of living and how freaking expensive everything is. That could be a confounding variable there. And a lot of the women that are presenting are on their second or third pregnancy, and so they are automatically excluded from the study. Another issue that we're seeing is that many of the women are presenting with a high BMI, and obesity has been shown to alter extracellular vesicle profiles. So we've been excluding women above a certain BMI as well. And then one of the biggest issues is that eligible participants just don't want to talk to the clinical researchers in general. So this project isn't the only one that's been having some recruitment issues. It's kind of across the board right now. And I mean, I get it, right? Like the first time you are pregnant, you are just slammed with so much information. You're overwhelmed with the fact that you're pregnant. You've got your doctors telling you stuff. You've got nurses telling you stuff. You've got your family maybe in your ear as well. And then you have somebody saying, hi, do you want to do research? And you're like, I know if I can take one thing off my plate, it's going to be this thing that I don't have to do. So I get it. But that does make it much harder for us to complete the study. So we're working with our clinical staff to see if there are ways that we can maybe increase their susceptibility or want to participate. So we're working on that. But the overarching kind of short-term goals with this project, the first is to identify stress-associated biomarkers during pregnancy. And this is kind of going into just generating data for a larger proposal to be able to fund a larger study. And that leads into what our long-term goals are, which is to conduct a study with a larger cohort of pregnant mothers. And then we would like to be able to separate out the types of trauma and PTSD, right? Are there differences in people who are experiencing a violent event or a sexual event or a natural disaster event, right? So all of these things may lead to different types of PTSD that we could then explore a little further. We also want to be able to compare EV and extracellular extracellular RNA profiles with control and trauma-exposed non-pregnant women as well. And then with this the kind of overarching long-term goal, as I kind of alluded to at the very beginning, is to be able to implement early intervention programs for people who are at risk of developing PTSD or who are currently suffering with PTSD and are not getting access to the help that they need to be able to have a better mental health state and therefore a better pregnancy. And then I would love to be able to conduct a longitudinal study that follows these women and then follows their children throughout childhood. Obviously, that's going to be a lot more challenging to do. Getting blood from kids is not something that most people are just willy-nilly wanting to do. So we'll see how that goes if it ever comes to fruition. So with that, I would like to thank my collaborators, Wilson Brown, who was my main collaborator on this project. He was also my mentor when I worked through my CTSI CERC fellowship. He recently left for Case Western. And then Alyssa and Ariana are the graduate students who were conducting all of the clinical interviews. Alyssa actually just defended her thesis. She was doing a project focusing on PTSD treatment for pregnant women, which, spoiler alert, there is no standard care for treating pregnant women with PTSD. Who would have thought? But she's staying on to continue doing our clinical interviews, so there's no disruption or change of hands with things. So we're really appreciative of that. And then our collaborators at McGee Women's Research Institute, Linda Paternity and Ashley Berg, who were instrumental in getting all of our collaborative agreements, material transfer agreements, and everything kind of situated. Helena Szynski, who is our kind of point person for all of our medical portion of the work. And then the two clinical staff right now, Erica and Kristen, who work through participant recruitment. And then our funding, this pilot project was funded by a Penn State faculty seed grant between Wilson Brown and I. As I mentioned, our community engaged research fellowship that I had, and then Helmet Health Foundation. And all of this work is being conducted through the Women's Health Innovation and Science Translational Lab at Behrend in collaboration with McGee Women's Research Institute. And then we're able to pull all of the electronic health records through the Mommy Biobank at McGee Women's Research Institute. So, and then thank you for your attention and attending. And I'll take any questions.
Thank you so much, Ashley. This was so interesting. We do have one comment in the chat. Kelsey says she wonders if there would be a recruitment difference in patients who go to an OBGYN versus a midwife. Do you have any thoughts about that?
Yeah, I am not sure. I've never personally interacted with a midwife. I've not had any experience with them. I know at McGee, the providers are either nurse practitioners or OBs. So I don't know what the difference would necessarily look like, but if you have any thoughts, I'm definitely open to it.
Yeah, I'm happy to chat. So I have two kiddos and have seen an OB for each of my pregnancies, and we had one midwife on practice, and I know it's more common in areas where midwives are available is one thing, and it's not like a shortage of care, for just like a midwife practice, to see patients, and they usually see them for a lot longer than your, 15-minute appointment that you get with your OB, and they can talk through a lot of the factors of pregnancy beyond, what the medical textbooks say, for example. So I can almost see it being a way that the midwives build rapport with you, the midwives build rapport with their patients, and so in turn you might be more likely to recruit them because there's just that higher level of trust, between the clinician and the patient, and then you as well. Of course, that's subject to availability of midwives and where you're recruiting, and so it might not be possible. But I thought it could just be interesting to look at.
Yeah, no, that's a really great point. I think it's also important to note that the way that the OB practice is set up here is that you rotate through all of the practitioners. So you don't even really get that chance to build a rapport with a specific OB. Like when I was pregnant with both of my kids, I saw pretty much every provider that was available because it was just, well, there's an opening on this day that works with your schedule. So this is who you're going to see. Hope you like them. kind of thing. So I'm sure it's very different if you have the opportunity to build that rapport with one specific person. And especially, right, given your point that you would have more than just your 15 minutes, like, hey, how you doing? Any problems? No? Okay, cool. See you later. Like that's very different than being able to talk through like what's actually going on with you.
Yeah, I had a very similar experience for sure. The other thought that I had as you were presenting was my first pregnancy started in 2019 and then 2020 happened and it was very traumatic
Yeah,
time.
I can
So
imagine.
it's probably good to be doing the study now where we're through the pandemic, but it would have been really interesting to do the study back in 2020 where, you started out with a normal pregnancy in a normal world, and then third trimester rolls around and life has changed completely. And I would have loved to see like the biomarker differences first trimester to third trimester at that period of time.
That would have been just amazing to have had set up at that point in time to be able to just perfectly capture that because I'm sure that would have been like such a crazy, crazy thing to see.
Yeah. Well, first we'll invent time travel and then we'll go back and do the study.
I don't want to go back. I don't want to go back.
Well, thank you so much. This has been a wonderful presentation.
Thank you.
Does anyone else have any questions that they would like to ask?
I have a question. First, I put in the chat, but I had no idea that we had a women's health innovation and science translational lab. So that's awesome. Um, and I would love to learn. I think all of us at CTS, would like to learn more about that so we can collaborate. I think I remember at the beginning you mentioned using the biomarkers to or using the EVs as a biomarker to identify issues, and then also treatment. And I was wondering, I know you're really early in your research, but have you identified what that might look like down the road? Would it be like a screener for women seeking care in the way that now there's a survey, but would there be some sort of like blood test that's done? And then if there are, for those who are deemed to be at risk, what kind of treatment options would you look for, both with the mom and the child?
Yeah, so in a perfect world, I would love if when you get your first trimester blood draw, they just take a little extra to separate out the extracellular vesicles and
Mhm.
look for a specific signature that might indicate that you're high risk for having some bad things associated with PTSD or trauma and things of that nature. And if you pop up as someone who has those profiles, then they can start mediating connections for you, right? Like, hey, I see that there might be something kind of flagging here. Let's get you in contact with a mental health provider that's in the hospital system and see, how we can start getting treatment for you to work through whatever it is, to hopefully increase your mental health and just avoid the bad things that could be on the horizon, right? So that could be, therapy. Obviously, like I mentioned earlier, we don't have a standard point of care for women experiencing PTSD during pregnancy, but maybe pulling from some of that literature that's out there and see, what might be a good approach. And that might be different for what type of trauma that person has experienced, right? What treatments they may have had in the past, and just exploring different treatment options that would work best for that specific individual. That's kind of like where my mind goes at this point in time. But yeah,
Thank you. This is really great work.
thank you. And yes, the Women's Health Innovation and Translational Lab is pretty new. We were calling it our Biomedical Translational Lab, but because of our partnership with McGee, we're like, this is the perfect way for us to position ourselves focused on women's health. And so that's kind of where we have pivoted and we're working to build it up a little bit more. So
That's awesome.
thank you.
Awesome.
Anybody else? Well, Ashley, this has been a fantastic presentation. I feel like your work is going to help with the stigma of mental health, especially in rural communities where, they might not want to talk about mental health, but if you do that blood draw that's happening anyway, we might be able to help more people through the PTSD. Are there any coping mechanisms or supports that would be offered to people who are found to have the PTSD biomarkers?
Currently, we don't have anything in place at this moment in time, but in the future, there are specific like cognitive behavioral therapy, like tactics that can be utilized to kind of help people. There's also like group therapy settings that some people might find more appealing than individual therapy settings. Um, there's a when that graduate student was working through her thesis, there were so many different approaches. And again, without there being like that standard point of care, it's really hard to say like what would be implemented first, but kind of drawing upon that would be kind of where I would go initially and see how things go.
Yeah. And I love that you use students too. That's why they're here, right?
Yeah.
Let's use them.
It's a great
um,
learning opportunity for them,
it
right? They have to
is. It
get their
is.
clinical hours in. And so this is a really awesome way for them to do that in a real world setting too, right? They're actually interacting with participants in a study, like with a faculty. So it's a really great experience.
That's really cool. Well, if nobody has any other questions, I do want to let you know that our next TSS is going to be on October 29th. Our speaker is Jessica Thompson, and she is going to talk about healthy homes and communities and improving lung cancer prevention for Appalachian women. So Dr. Thompson is part of our CERC program, so they will be presenting, like I said, October 29th. All the materials from today will be sent out. There will also be a survey, so please complete that if you can. It helps us make these sessions so much better. So with that, I would like to thank you for your time this afternoon, and thank you again, Ashley.
Thanks for having me.